I was trained by really fantastic scientists who told me that you should always tell people why what you work on is an important problem. And I realized that, at the same time that organizations, depending on their mission, have different agendas or kind of different priorities. But, what I tell people about TB and the work that we do is that, there's no guarantee that TB is a solved problem here in the US or anywhere. And so I think it always is valuable to continue to think about investing in the problem. I also think that, there's so much that we can learn by understanding how to manipulate the immune system. So when I'm talking to somebody in cancer, for example, I tell them, I study TB, but what I also contribute to that kind of conversation is that I am developing ways to modulate the immune system. And everybody wants to do that right now. Right. To people in different fields or in different kind of sectors of the biotech industry, I just say, I figure out ways to modulate the immune system. I use TB as my core problem of interest, but the general paradigms that we learn could be applied across a whole host of different systems. And we've shown that. And so to me, my partnership with industry is, when I think about who also wants to target these immune cells, they might want to be targeting them in a tumor, but what can we learn from each other about what have we tried, what have you tried, that might be either useful or a great insight that I could provide to somebody else is like, we tried that, it didn't work, let's try something else. So being able to barter in the higher level of kind of knowledge where maybe you're not necessarily invested in altering the immune system in the way to kill bacteria, but you're interested in altering the immune system, and I can talk that language, no problem. So when I think about partnering with industry, I think about how can we learn to modulate these innate immune cells specifically? And I kind of, if you want to talk about TB as well, I'm happy to do that. But I think, what I've learned is that you meet people where they are and you find mutually aligned interests, and a mutually aligned interest that we for certainly share with a lot of the pharmaceutical industry is how to modulate the immune system. And I think what we learn in our system is directly beneficial to what they're doing because, there's been a long-held dogma, what I'll say about these particular cells of the innate immune system that we study. And not to toot our own horns, but I'm very proud of my lab because, we'll complete 3 years of having my lab in July. And I've never taken an immunology class, but I do own immunology textbooks. And there's a very famous line about the immune cells that we study. And everybody says these cells do this. And, in 3 years, we disproved that. So I think that even though we study TB, we also study the immune system. And I think we are active contributors in that space. And so that's why I think that what we do is valuable. And you just have to, if you're not interested in TB, just read the other figures because there's still something very much to be learned there. So there's this textbook model for the cells of the innate immune system that we study. We study these immune cells called macrophages. And if you read many textbooks or reviews on these cells, one of the things that you'll often come across is this concept that these cells are plastic and polarizable. So to talk about the polarizable piece first, these cells have been characterized as either inflammatory or anti-inflammatory. So you have these two poles, but the plastic piece comes in where the idea has always been that you can go from the anti-inflammatory state to the inflammatory state and vice versa. Okay, and here's a lesson in how to not be an advisor. Because I remember when I asked one of my first students, I said, try this experiment. We don't know what's going to happen. And so the student designed this really, really very beautifully designed experiment. And then he said, oh, I did this and it didn't happen. And I said, I think you did the experiment wrong. And I was like, I regret that to this day because I have this bet in my lab that if I say, like, if I propose an experiment that's too crazy, or if you just like fundamentally disagree with an experiment that I think you should do, the bet is that I have to come in and do it. And so he said, well, you know, I'm calling you on that. And so I had to come in and I did the experiment. I said, whoa, like I did it myself. And I was like, I saw the exact same thing. And I was like, wait, like the textbook model sets up this idea that we should just be able to take these cells and give them this and they'll convert into this state. Actually turns out to not be the case, but it's a lot more complicated than even that, because the way that we actually characterize whether a cell is in an inflammatory state or an anti-inflammatory state is like very narrow. You could think there's like 20,000 proteins, but maybe we use 5 proteins to characterize that you're in an inflammatory state or an anti-inflammatory state. That's like, you know, in the era of systems biology, that's heresy. And so, you know, so when we actually said, okay, what happens when we look at the systems level, what we actually ended up showing is that, you know, the model is somewhat true and somewhat wrong. So in fact, what happens is that once you become inflammatory, there are just a set of genes that you just cannot ever turn back on. And so that's why the cells are quote unquote stuck. So if you only measured those genes, you would say the model is flawed. The textbook model is flawed. But if you instead looked at these other 12 genes, you would say the model is correct. And so that's kind of the beautiful thing about the system, is that what we've shown is that actually the model, the textbook model falls short of what the true biology says. But what's super cool there for us is that what we think is that the protein that we've identified that actually targets these genes that don't change is actually a huge pharmacological target for chronic inflammation. Because what happens is that these cells get stuck in the inflammatory state, and many of the canonical responses that you would want to turn on kind of an anti-inflammatory state get blocked. But now that we figured out what protein it is that controls this switch, so to speak, now we can come up with now we have a drug target. Now we can begin to figure out how do we target this such that we can relax the ability for the cells to convert back into this anti-inflammatory state.